Hello, everyone, and welcome to our ATRN seminar. The speaker today is Yusuke Imoto of the Kyoto University, and the title of his talk is Single-Cell Trajectory Analysis with VMapper.
Thank you for kind introduction. So good morning, everyone in Europe, and good afternoon, everyone in Asia. So it is a great honor to be here today at the Applied Algebraic Topology Research Network Seminar. So my name is Yusuke Imoto from Kyoto University. So I'm an associate professor of the Professor Yasuhiro Laboratory. So I am delighted to have the opportunity to present my work on single-cell trajectory analysis with VMapper. So at the beginning, I would like to introduce my institute, ASHB. So ASHB is one of the world premier international research center initiative called WPI at Kyoto University, which was established in 2018. The target of ASHB is human biology, and the main question is what key biological traits make us human? And how can knowing these lead us to better cures for disease? So actually mainly consists of the biological teams, but includes the two math group and one ethics group. The one of the mathematical group is a math data analysis group organized by Professor Hiraoka. And this group has three postdoc and me. So especially Miha gave a talk about the topological node to Beck in this seminar last month. The goal of this study is to understand both the shape and flow of the cell differentiation using a single-cell RNA-seq data. The cell differentiation is the process of the cell specializing to the other cell types. In this process, cells change their gene expression level. The gene expression is the process of the gene synthesizing RNA and protein. Generally, mammal has 20,000 genes. So therefore, the cell is characterized as the amount of the RNA or proteins. So that is, so we can regard the cell as a vector and the cell differentiation as a trajectory in a gene expression space, which is a 20,000 dimensions. The single-cell RNA-seq data is a data measuring the amount of the RNAs for entire genes at the single-cell level. That is, the single-cell RNA-seq data is a point cloud in the 20,000 dimensional space. So we aim to reconstruct the trajectory of the cell differentiation, which is the topology of the point cloud and its dynamics using the topological data analysis. So here we must note that the characteristics of the single-cell RNA-seq data, such as high dimensionality, containing a technical noise, non-time series, and the high experimental cost. As previous studies of the trajectory inference using the single-cell RNA-seq data, there are mainly two types of the methods. One is the pseudo-time analysis, which is a tree-based method that approximates a point clouds by the minimum spanning tree. So this method can infer the pseudo-time, which is a flow, by setting the starting point, whereas cannot detect the topological information. For example, in this figure, so we set the starting point to here. So then number 0, so next is number 1, next is number 2, number 3, and the others are numbers 4. So then, so this number corresponds to the time, should time of the data of the this minimum spanning tree. So another is the mapper, which is a topology-based method that can detect the topology information like a plasticity, cyclicity of the cell differentiation, but so it cannot infer the flow. So to resolve this trade-off, so we utilize the velocity information. So in single-cell RNA-seq data analysis, there is a groundbreaking approach that can infer the velocity of the gene expression from the single-time single-cell RNA-seq data, called RNA velocity, which are introduced in the next page. So accordingly, so we can consider the data analysis platform whose inputs are point cloud and velocity. Thus, we have developed a velocity mapper called VMapper by embedding the velocity into the mapper graph. The VMapper represents a topology and flow simultaneously by a directed graph, which is called VMapper graph. We aim to apply the VMapper to the single-cell seq data and to understand the cell differentiation mechanism. So here I would like to explain the RNA velocity briefly. So RNA velocity considers the variation of the RNA molecules, which is unspliced and spliced messenger RNAs. So in the cell, a gene transcripts the unspliced messenger RNA U called the intron, and it changes to the spliced messenger RNA S, so called the exon. So furthermore, the exon translated to the protein. So here, the amount of the spliced RNA corresponds to the gene expression value of the single-cell RNA-seq data. The RNA velocity models the gene expression processes by this OD model. So since S means the gene expression level of in the single-cell RNA-seq data, so then the right-hand side of the second equation expresses the velocity of the gene expression level. So moreover, in the single-cell RNA-seq platform, the amount of the U and S are observable. So therefore, we can estimate the velocity by evaluating the parameters using the statistical methods like a regression. So because the single-cell RNA sequence technology cannot extract the time series data, so RNA velocity is widely used to infer the dynamics of the cell differentiation. So the contents of this talk consists of the three parts. So first, we introduce a mapper algorithm. Second, we introduce the VMapper algorithm. Finally, we introduce a single-cell trajectory analysis with VMapper. In the actual single-cell RNA-seq data analysis, there are some practical problems such as the curse of dimensionality, which is a statistical problem when we analyze high-dimensional noisy data, and the interpretation problem of the complex flow on the VMapper graph. So we will introduce such problems and their resolutions with high-dimensional statistics theory and Hodge decomposition. So now let's introduce a mapper algorithm. So mapper constructs topology based on the idea of the knob. So let X be a topological space in Euclidean space. So a cover of the X is a collection of the non-empty subsets phi of X, whose union is a fold space X. So then we define the knob N of phi as all non-empty subcollections whose set have a non-empty common intersection. For example, the X is a square with a hole, and we make the cover phi by subsets consisting of each side of the square, B1, B2, B3, B4. So then the knob of the graph, knob is a graph whose nodes correspond to the subset, and edges correspond to the overlap of the subsets. So under these settings, the following knob theorem holds. If phi is a finite cover consisting of the closed, convex, and non-empty subsets, then the knob N of phi and X have the same homotopy type. So then homotopy type of the X and N of phi are the same. Mapper constructs the cover from the data and represent the topology by its knob. Now I introduce the mapper algorithm. The mapper algorithm has four processes. So first, we map the original data to the low-dimensional space. So let bold X be the point cloud in D-dimensional space. Then we map point cloud X into the P-dimensional space by the filter function F. Here, the blue box means a parameter of mapper. So we have a point cloud projected into the low-dimensional space. Second, we make cover of the point cloud projected into the low-dimensional space. So let U be a cover of the point cloud f of x projected by the filter function f. So in addition, we construct a cover of the original point cloud U of f. So by putting the cover back into the high dimensional space. Third, we decompose the component of the cover of the original point cloud by the clustering method. So using clustering method for each component of U of the cover, we decompose F inverse of U into the finite clusters. So we construct a new cover by the set of the clusters. So finally, we construct the nerve. So here we only consider the graph corresponding to the one-dimensional simple complex. So we construct the mapper graph whose nodes are assigned to the indices of the clusters and whose edges are defined between the overlapping clusters. So here n is a number of the clusters and lambda is a bijection from the vertex set to the cover made by cluster. So we wrap up the algorithm of the mapper. So mapper defines a cover of the original point cloud by a mapping, covering, and clustering processes. Then mapper constructs a mapper graph following the definition of the nerve. So mapper has three parameters: filter function, cover, and clustering method. So regarding the parameter study of the mapper, please see the appendix of my paper. So as a remark, so we can also consider the high-dimensional mapper graph corresponding to the simple complex or hypergraph by supposing the interactions among the three or more clusters. However, we exclusively only use this graph because this study considers the flow of on the mapper graph. So now I showed a simple example. The input is a point cloud forming the standing cat. So we apply the mapper to the cat data. So we set the parameters as a filter function is an identity function. The cover is a cube divided into the 8 parts along each axis with 50% overlap. And the clustering is a DB scan. The right figure is a mapper graph that visualizes the topological structure of the cat, which is a body and some branches corresponding to the foot, tail, and ears. So, by the way, so he is my cat, mimicking these input and output. So since he is a talented applied mathematician, so he can compress his body into the two-dimensional space without without mapper. So by the way, so I introduced application of the mapper. So mapper has been applied in the many research fields, including the life science, neuroscience, molecular biology, chemistry, and economics. So recently, I have applied mapper to the rock fracture structure data to evaluate the permeability of the flow in fracture structures with Professor Suzuki's laboratory at the Tohoku University. So next I introduce the proposed method, VMapper. So VMapper is the extension of the mapper using the velocity data, which is additional input data, and the VMapper graph represents the topology and the flow by a weighted directed graph. So we explain the embedding of the velocity into the mapper graph. So recall that the node of the mapper graph consists of the clusters of the point cloud, and the edges are drawn between the clusters with interaction. So we denote the ith edge and its connected nodes in mapper graph as EI, I1, I2 respectively. So there exist corresponding to clusters and their interaction. So lambda is a map from the node indices to the cluster. So the B mapper determines the direction and weight of the edges in the mapper graph by the following procedures. So first, we compute the center of the clusters and the relative position vector Ri between the centers. Second, we compute the mean velocity Ui for the points in the intersection of the clusters. So finally, we compute the weight Wi of ith by linearly projecting the mean velocity Ui into the relative position vector Ri. So then that we define the direction of the edges by the sign of the corresponding weight. So in the weight is a positive, so the direction is from I1 to I2. So in contrast with negative, the direction is I2 to I1. So if the weight is 0, so we eliminate the noise of the edge. The absolute value of the weight shows the flux of flow. So before moving to the demonstration of the VMapper, I would like to introduce an adaptive hypercube cover, which is a cover used in my paper. So generally, a hypercube cover is very used as a cover of the mapper. So the hypercube cover is a cover dividing the data evenly. So the hypercube cover is easy to implement, but it is weak against the data sparseness. So we propose the adaptive hypercube cover, which is a set of the hypercubes in which the number of the samples in each hypercube is as equal as possible. So since the size of the hypercube is automatically determined from the input data, so the number of the parameters of the adaptive hypercube is the same as that of the non-adaptive one. So I show an example which defined from the previous cat data because there is no velocity in the cat. So this is an OD system known as a Lorentz equation. So Lorentz equation has an attractor that orbit around the 2 points. So we generated the input data from the trajectory of the Lorentz equation with constant time increments. So we set the parameter as a theta function is a two-dimensional principal component analysis, and the cover is an adaptive hypercube cover with a 50% overlap, and the clustering is a DB scan. The right figure is a B-mapper graph, and we can identify the direction of the orbit in the attractor from the direction of the edges. So note that we couldn't obtain the true topological structure when we used the non-adaptive cover, but otherwise the same parameters due to the data sparseness. So in the third section, we applied the VMapper to the real single-cell data in order to infer the trajectory of the cell differentiation. So we use a single-cell data for pancreatic endocrine cell. So this data contains the 3,700 cells and the 28,000 genes. So this data has 8 cell types, and a previous study showed that endocrine cell differentiated into the 4 cell types of the endocrine, alpha, beta, epsilon, and delta at the last stage. However, it is an open problem of which cell types these mature and clean cells are differentiated from. In this study focusing on the last stage, we use the pre-end clean and these four types of the end clean cells, which consist of the about the 1,900 cells. So this is BMapper graph of the mouse and clean cell data. The colors show the main cell types in each cluster. So you can observe the sphere-like structure. So does this mean that all endocrine cells are differentiated from the one another on the sphere? So unfortunately, it is not. It is false. So why did we obtain such structure? So this is due to the curse of dimensionality. The curse of dimensionality is a statistical problem when analyzing the noisy high-dimensional data. For example, let X true J be the true data with a deep feature and the XJ be the observed data containing the noise for each feature. So then the curse of dimensionality is an inconsistency problem, inconsistency between the structures obtained from data analysis of the true and observed data. So for example, the distance or sample statistics among the observed data may be overestimated rather than those of true data. The mapper is also affected by the curse of dimensionality because the mapper has a filtering and clustering procedures in the original high-dimensional space. So we demonstrated the effect of the curse of dimensionality of mapper using the double 8-figure structure data. So this example demonstrated that the topological structure of a mapper are destroyed as the dimension increases. So as an aside, percent homology also cannot escape the curse of dimensionality. So for example, so we prepare the two cycle data as a true data. The true percent diagram has a two generator. So we embed the true data into the D-dimensional space and add the tiny noise for each component. So then the number of the generator increases as the dimension increases. In the 10,000 dimension case, we cannot identify the true topological structures any longer. So regarding this observation, Mr. Liu, who is a member of our laboratory, mathematically proved the curse of dimensionality in percent diagrams for check and Lip's case. So moreover, he showed that utilizing the normalized PCA mitigates the curse of dimensionality in percent diagrams. So please see his preprint for detail. So in the case of the single-cell RNA-seq data, we have developed a noise reduction method based on the high-dimensional statistics called record. The record stands for the resolution of the curse of dimensionality. So the high-dimensional statistics is a research field that investigates the asymptotic behavior of statistics when both sample size and dimension increase. So record first normalize the single-cell RNA-seq data so that noise variance equal to 1. Next, record identifies the essential and noise part based on the singular value decomposition. So finally, the noise part is eliminated and eigenvalues of the essential part are modified by the high-dimensional statistics theory proposed by the Professor Yata and Aoshima in Tsukuba University. The important property of the record and record are accurate, fast, wide applicability, parameter-free, and a priori validation. So we summarize the procedure of the VMapper for single-cell RNA-seq data. So we first apply the record to original single-cell RNA-seq data to resolve the curse of dimensionality. We next apply the RNA velocity method to the single-cell RNA-seq data to obtain the velocity data. So then we apply the VMapper to the denoised single-cell RNA-seq data and RNA velocity data. We set the parameter as a theta function is a three-dimensional multi-dimensional scaling, and the cover is adaptive hypercube cover with 50% overlap, and the clustering is a single linkage with the first gap detection, so which is a standard parameter-free clustering method in Mapper. This figure the VMapper graph with decode. The VMapper graph consists of the 3,700 nodes and the 70,000 edges. So compared with the VMapper graph without record, so this VMapper graph represents the fine topological structure, which leads to the resolution of the curse of dimensionality. So moreover, compared with the dimensionality reduction by MDS, we clearly observe the connectivity among the cell types. So furthermore, this is a previous analysis using the UMAP and RNA velocity. So UMAP is a nonlinear dimensionality reduction method based on the manifold learning. So compared with the previous results, the connectivity among the cell types of the VMapper graph are well interpretable. So from the VMapper graph, we construct this diagram showing the topology of the cell differentiation. Next, we want to evaluate the flow of the cell differentiation using the directed to edge information on the VMapper graph. So however, this graph is so complex that we cannot infer the developmental flow just by observing the directed edges. So we simplify the flow using the Hodge decomposition. So we suppose the cochain complex, for example, the sequence of Hilbert spaces connected by the co-boundary operators. So then the Hodge decomposition decomposes the space to the three orthogonal direct summands. So in vector calculus, so the Hodge decomposition leads to the Helmholtz decomposition by setting the co-boundary operators as the gradient and adjoint of the curve. The Helmholtz decomposition decomposes the vector field into the gradient flow, harmonic flow, and the rotational flow. In the cell differentiation, it is considered that the gradient, harmonic, and rotational flow corresponding to the development, plasticity, and cell cycle, respectively. So therefore, we can infer the developmental flow of the cell differentiation by extracting the gradient flow. So moreover, the Hodge potential visually helps to understand the gradient flow because the gradient flow is represented as the gradient of the Hodge potential. So however, the computational cost of the Hodge decomposition depends on the dimension because in the calculation, we have to compute the high dimensional Poisson equation. So then it is not reasonable to use the Hodge decomposition as it is. So we use the combinatorial Hodge decomposition, which is a Hodge decomposition on a simplification complex or graph. So we explain the combinatorial Hodge decomposition on graph. So let C be the weighted directed graph without self-loop. So let T be the set of the triangle on a graph. The gradient of the edges is defined as a difference in the value of the connected nodes. So the curve is defined as the summation of the value of the connected edges. So then the graph and curve satisfy the condition of the co-boundary operator. So combinatorial Hodge decomposition decomposes the weight vector of the graph into the three vectors. The graph reconstructed by the decomposed weight vectors creates the three new graphs. For example, this is a decomposed, this graph is decomposed into the these graphs by the combinatorial Hodge decomposition. The gradient shows a cyclic graph. The rotation shows a cyclic graph on the triangle, and the harmonic is a remaining component. So the blue number shows the Hodge potential of node. So from the graduation of the numbers, Hodge potential, we can consider the flow. We can regard the flow. So we apply the combinatorial Hodge decomposition into the B-mapper graph and colored by the Hodge potential. Since the gradient of the Hodge potential shows the flow of the development in a cell differentiation, we can determine the flow among the cell types. So consequently, we conclude the topology and flow of the cell differentiation for the mass and clean cells by this diagram. So now we are applying the BMapper to real immune cell data and biologically verify it. So we summarize my talk. So BMapper is a mapper-based topological data analysis method describing the topology and flow from the high-dimensional data with velocity. So mapper and BMapper suffer a curse of dimensionality. So the combinatorial Hodge decomposition describes the developmental flow of the cell differentiation. The combination of the BMapper record and the combinatorial Hodge decomposition for single-cell RNA-seq data revealed complex dynamics of the cell differentiation at the late stage of the mouse pancreatic endocrine cell. The code of BMapper is available from my GitHub. So these are references of this talk. Please confirm it on YouTube if you are interested. So that's it. Thank you for kind attention.
Thank you so much. I would ask everyone to unmute themselves now and give a round of applause for our speaker. And before we get to questions, I have to say that that's a beautiful cat.
Thank you very much.
Questions. Feel free to raise your hand or just unmute yourself and ask.
I'll start with the question. So the if I understand correctly, the Hodge Laplacian part that still gives you a arrow on each edge, right? And then is it sort of a user-informed process to go from the vectors on each edge to then produce this simpler model where you had only 6 nodes with the arrows? Like I'm thinking of the picture with the complicated network and then the actual gene regulatory network. Yeah,
gene
here.
regulatory network. Yes.
Oh, no.
Sorry.
Go to the slide with the data. Yeah, here, this one. So is it sort of a user-informed step to go from the figure on the left to the figure on the bottom right?
Yes. From this figure, so we estimated from the gradient of the Hodge potential, so we estimated the direction of the cell types among the cell types. So can you make sure?
Right.
So
right.
then,
But the Hodge allows you to, like the figure on the left, you might have many arrows pointing in different directions locally, but then the Hodge Laplacian will sort of smooth that out.
Yes.
I see.
So I cannot determine the flow from the by observing the all edges. So then then to simplify it, so we make no we represent the flow, gradient flow by the color
Right.
of the Hodge potential.
Ah, gotcha. Gotcha. I see.
Yes. So red to yellow. So this is direction. In case of the graduation from the color of gradation of color, so we can determine the direction of the flow.
It's a very nice application of the Hodge machinery.
Yeah. Thank you.
More questions?
I couldn't hear.
Sorry, can I ask one question?
Okay.
Oh, thank you for the talk. I think you have some parameters to tune for
Yes.
the VMappers
Yes.
to get some results. And what kind of the criteria used to determine that you got the good good mapper results or so on? So what kind of things you consider when you judge whether your resulting mapper graph is successful or not?
Thank you. So yeah, so because there are three parameters, so it is difficult to determine the parameter. Determine the good parameters and evaluate it. So, but so yeah, especially so the filter function cannot destroy the structure, especially the distance structure of the original data. So then the MDS or PCA is well used to the mapper. And so cover is determine the resolution of the resolution of the data. It means that so for example, the division, the number of the division is too small, so we cannot we can only see the, for example, very small nodes, so it cannot construct the topology. So on the other hand, if the number of the division is too large, so all points the cluster cannot have an intersection, so then it is also has no it does not have the it is also has no it does not have the topology. So then practically we check many parameters, and so we found the stable area. For example, we check the 23, 24, 25, 26, 27. So yeah, if the topological structure is stable around the 27, it's good. I determined. So yeah, this is the clustering is used single linkage or DB scan in general. So this is also cannot destroy the distance structure. So then, we summarize the parameters selection. So the beta function and clusterings these have a common method, and so cover determines the resolution. So then we determine the many parameter. We check the many parameter, and we determine it is good or not.
Okay, thank you. So you changed the covers, you changed the parameters, and
Yes.
find some stable parts of those
Yes.
results.
Yes.
Yeah, thank you very much.
So we have a question in the chat window. Could you please repeat the definition of a Hodge potential?
Okay, thank you very much. Sorry. Yes. Uh, どっちにしましょうか。Yeah. For example, the flow is decomposed to the hot gradient flow and harmonic flow and the rotational flow by the Hodge decomposition or Helmholtz decomposition. The gradient flow is represented as the potential, the gradient of the potential. So this potential is called the Hodge potential. So then the potential shows the scalar value, potentially the scalar value, and its gradient represents this gradient flow. So then, yeah, phi is a definition. So in the graph case, then this is a Hodge potential assigned to each node, and the difference between the Hodge potential is a flow of edges. Okay. I didn't know it's. Yes. Yeah. Generally, this is potential flow. This is normal potential. But in case of in terms of the Hodge decomposition, we say the Hodge potential.
Okay, wonderful. So I think we can take one more questions and then we're gonna take questions offline. Or if people are shy, then we're just gonna take more questions offline. Any for the record questions for Yusuke? Okay, so in that case, thank you so much again for your talk. I will stop the recording now and then the remaining questions can be offline.
Thank you.
Thank you.